Good day, Ma'am Fadge. I am Chelsie Marrie Laquio, and my presentation is about the isolation and identification of Klebsiella pneumoniae from an unknown bacterial sample. The main goal of this experiment was to isolate, characterize, and identify Klebsiella pneumoniae through microscopic observation, cultural characteristics, and biochemical testing. Klebsiella pneumoniae is a gram-negative, encapsulated, and non-motile bacillus that belongs to the Enterobacteriaceae family. It is part of the normal intestinal flora, but it can become pathogenic, causing pneumonia, urinary tract infections, septicemia, and wound infections. Its most distinctive feature is its capsule, which makes the colonies appear mucoid and helps the organisms resist phagocytosis. In the laboratory, K. pneumoniae can be identified using selective and differential media such as McConkey and EMB-agar, and through biochemical profiling including IMVIC, TSI, and sugar fermentation tests. Now moving on to the methods, I performed the procedure over 4 days. On day 1, I began with a gram stain, which showed gram-negative rods. I then inoculated the sample onto McConkey and EMB-agar, and then incubated the plates at 35 to 37 degrees Celsius for 24 hours. On day 2, I observed large mucoid pink colonies on McConkey-agar, which is typical of lactose fermenters, and purple colonies on EMB-agar without a metallic sheen. I then subcultured the organism to different biochemical tests such as IMVIC, motility, TSI, PAD, urease amino acid decarboxylase, sugar fermentation, and OF glucose tests. And then on day 3, I recorded the biochemical reactions, performed the string test, and inoculated the organisms onto XLD and blood agar plates. I also re-incubated the urease broth. Now, here is a summary table of all the biochemical tests I performed, which will be further discussed later on results and discussion. And then lastly, on day 4, XLD colonies were yellow, BAP colonies were non-hemolytic, and the urease broth still remained negative after 48 hours of incubation. Now, moving on to the results and discussion. Under the microscope, I observed gram-negative rods, indicating that the organism belongs to the enteric bacilli group, which was consistent with the characteristics of Klebsiella pneumoniae. On McConkey-agar again, I observed large pink and mucoid colonies, which confirmed lactose fermentation. On EMB-agar, colonies appeared purple without a metallic sheen, which differentiates Klebsiella from E. coli. On XLD-agar, colonies were pale yellow, showing sugar fermentation. On blood-agar, colonies were non-hemolytic, meaning the organism does not produce hemolysins. Now, moving on to the biochemical tubes, TSI had A over A with gas, which means it ferments glucose, lactose, and sucrose with gas production, but no hydrogen sulfide. On indole, it was negative. The organism lacks the enzyme tryptophanase and cannot convert tryptophan into indole. And then methyl red was negative. It does not use the mixed acid fermentation pathway, so no strong acid end products were formed. And then Vogg's Proskauer was positive, which means it produces acetone via the 2,3-butanediol fermentation pathway. And then lastly, citrate was positive. It can use citrates as a sole carbon source, which turned the medium blue. Now for the other biochemical tubes, OF glucose was fermentative because both tubes turned yellow, indicating facultative anaerobic metabolism. PAD was negative. It does not deaminate phenylalanine, which differentiates it from Proteus species. LIA was positive. It decarboxylates lysine, producing a purple color, and then the motility was negative, which means the organism is non-motile and lacks flagella. And then lastly, string test using a loop, positive because a viscous string formed, confirming capsule presence. Now, one interesting point in my results was the negative urease test. While Klebsiella pneumoniae is usually urease positive, enzyme production can vary among strains sometimes. Some strains show slower urease activity, requiring at least 72 hours for a complete color change. Now, there are possible reasons for the false negative results include a small inoculum, a short incubation time, which was my case, slightly acidic pH of the medium, and strain-specific variability. Therefore, even though my urease test was negative after 48 hours, the result still aligns with Klebsiella pneumoniae when considered with the other confirmatory tests such as the IMVIC, which was negative, positive, the TSI, which is A over A with gas, and non-motility. To summarize, the biochemical pattern I obtained in IMVIC was negative, negative, positive, positive, non-motile, lactose fermenter, lysine positive, and TSI A/A with gas is consistent with Klebsiella pneumoniae. In conclusion, the unknown gram-negative bacillus was identified as Klebsiella pneumoniae based on its morphology, cultural characteristics, and biochemical profile. The negative urease result was most likely due to strain variability or slow urease expression. That is why I recommend the following. First is to extend urease incubation up to 72 hours to detect slow urease activity, which is what I failed in my part as well. Then second is perform antibiotic susceptibility testing, since Klebsiella pneumoniae is known for multi-drug resistant. Now these two are the things I wasn't able to perform due to limited time and resources last week. Now, once again, the bacteria identified is Klebsiella pneumoniae. And that is all, Mag Faj. Thank you so much.